Everyone, and welcome. Thank you all for joining us today. I'm Julien Gillet-Daubin, Director of Capital Study in France. It's a pleasure to have you with us for this presentation. Today, we are going to dive into the world of Apmonia Therapeutics. We will explore their innovative approach in oncology and the exciting development they are bringing to the biotechnology landscape. Our session today will include about 10 minutes of presentation, followed by 20 minutes dedicated to your question and exchanges. So please feel free to submit your question in writing via the chat just below the video throughout the session, and we'll be happy to answer those questions after Albin's presentation. So without further ado, let's get started with a presentation of the management team of Apmonia. Maybe Albin, I'll leave you the floor.
Yeah, sure. Hello, everyone. Thank you for joining us today. So I'm Albin. I'm the CEO and co-founder of Apmonia Therapeutics. So briefly, a few words about myself. I hold a PhD in chemistry, and later I trained in business at HEC in Paris. I spent over 15 years in translational drug development, including as a project manager in the French TTO before I co-founded Apmonia now already 6 years ago.
Stéphane, maybe?
Yeah. Welcome everyone, and thank you for joining this webinar. I'm Stéphane Dedieu, a full professor at the University of France, where I had a CNRS research team of about 40 people. My research focus on deciphering the functional interaction between the extracellular matrix and the cells, especially in the field of oncology, and also in developing new strategies to target the non-cellular component of the tumor microenvironment. I'm a co-founder of Apmonia Therapeutics with Albin and Abderrahim, and co-inventor with Albin of Tax2 technology. I'm involved in the company as chairman of the Scientific and Clinical Advisory Board, and also as a scientific advisor to guide the R&D activity.
Okay. Thank you very much, Stéphane. Well, I'm Abderrahim Lashgar, co-founder of Apmonia Therapeutics with my friends. And concerning my background, I'm immunologist by background. I was involved for more than 25 years in biotech industry, from drug discovery up to phase 2 clinical trials with the proof of concept in patients. And I'm a serial entrepreneur. I'm also a member of the board of the company and strategic advisor for the management team.
Thank you very much. Albin, maybe you can start the presentation.
Yes, sure. So, yeah, perfect. So, yeah, thank you once again for being here today. So, at Apmonia Therapeutics, our mission is to transform the treatment of solid tumors by targeting the tumor microenvironment, and more specifically, the extracellular matrix, or ECM, to improve the survival outcomes for millions of patients. So we are based in Reims, not far from Paris. And Apmonia is originally a spin-off from the French National Center for Scientific Research, the CNRS, and the company was incorporated in 2019. Since then, we've received prestigious awards, including iLab, iNov, EY Entrepreneur of the Year, and the EIC Accelerator, which is the most competitive European funding for deep tech companies. We've also secured over 11 million euros in seed funding, and we are now preparing to enter clinical development. Cancer kills over 10 million people every year, and 90% of cancers are solid tumors, which are notoriously resistant to current therapies. A key reason is the extracellular matrix. It's a dense protein network that shields tumors, supports metastasis, and blocks immune responses. So targeting the ECM is an emerging and promising strategy with a projected $10 billion market by 2030. Apmonia has developed a proprietary discovery platform combining computational modeling and wet lab validation to design small peptides that disrupt tumor ECM interactions. So understanding the protein-protein interface allows us to engineer small peptides that bind to specific sites on ECM proteins and block the protein interactions that are known to promote the tumor aggressiveness. And these peptides are engineered at Apmonia Therapeutics to be highly potent, selective, and to avoid the off-target effects that are seen with other modalities. So we've built a pipeline of ECM targeting peptides with strong proof of concept in multiple solid tumors, including ovarian cancer, which is one of the deadliest cancers, and our first clinical application. Our candidates are protected by 4 patent families, and we collaborate with top academic teams. Our most advanced candidate is TAX2. It's a first-in-class cyclic peptide targeting thromboxanin 1 or TSP1, which is a glycoprotein from the ECM. And TAX2 is nearing a first-in-human clinical trial whose success will validate the whole platform. TSP1 is overexpressed in many solid tumors and drives the progression via its interaction with the CD47 receptor. We built the first all-atom model of this interaction and designed TAX2 to block CD47 activation by TSP1. And this is a new and safer way to reprogram the tumor microenvironment. TAX2 peptide works through 2 synergistic mechanisms. First, it activates the T-cell immune responses to attack cancer cells. And second, it also inhibits the tumor vascularization, promoting necrosis and tumor regression. TAX2 is now clinical trial ready. So the GMP manufacturing and GLP analytics are completed. We got strong preclinical data that shows efficacy and non-toxicity in 15 animal cancer models, including ovarian cancer, but also pancreatic cancer or glioblastoma, with excellent PK and biodistribution. The toxicology studies are done and they confirm a favorable safety profile. And last, our clinical strategy has been endorsed by the European regulators. Our first target is ovarian cancer. It's an orphan disease with 300,000 patients and no less than a 50% 5-year mortality rate, with extremely limited therapies. We aim to treat 10 to 50% of patients who are not responsive to current therapies, representing a 1 billion euros revenue opportunity within 5 years post-approval. TAX2 offers a unique mechanism of action. Unlike PARP inhibitors that are used in ovarian cancer, it's not limited to genetic subtypes. Unlike other CD47 targeting agents, it avoids severe side effects. And unlike antibodies, our peptides are more selective, safer, and also cost-effective. So by developing potent and selective peptides against the tumor ECM, we are pioneering a new therapeutic class and opening a whole new market focused on modifying the tumor microenvironment. Our model is to out-license candidates after the clinical proof of concept, generating up to 750 million euros in upfront and milestone payments based on recent deals. We are already in discussion with major pharma players, including, as way of example, BMS, Merck, and Mitsubishi Tanabe. And our long-term vision is to become the leader company in ECM therapeutics, with a diversified pipeline and a clear path to strategic exit, as seen in recent multi-billion deals for CD47 targeting agents, such as the acquisition of the biotech company 47 by Gilead. They had 1 compound in phase 1 for a $5 billion deal. Or another example is the takeover of Trillium Therapeutics by Pfizer for over $2 billion. Our go-to-market strategy is now to de-risk and validate our platform. So TAX2 will soon enter a first-in-human trial to obtain the clinical proof of concept in ovarian cancer by late 2027, and then reach an out-licensing agreement. In the meantime, we are also advancing other leads in oncology and enhancing our computational tools to further improve the platform. Our Series A will fund the phase 1 and phase 2A trial and support the pipeline expansion. We've already secured over 10 million euros from top-tier investors. 4 VCs, Capital Grand Est, Finovam, Noshak, and Sprim Global Investments. 1 venture philanthropy foundation, the Fournier Majoie Foundation. It's a Belgium-based foundation, and they only invest in oncology company. The EIC Fund, which is the investment arm of the European Investment Bank, will also be part of the syndicate, alongside Angel Santé, which is the leading European life sciences angel network. And besides, we've also raised over 6 million euros in non-dilutive funding, mostly in grants. And now we are opening the round to you and capital cell investors, a unique opportunity to join a high-impact, high-potential biotech company. So I co-founded the company with my partners Stefan and Abderrahim. We bring deep expertise in ECM biology and biotech entrepreneurship. And we built a team of 15 people with expertise both in early discovery, including bioinformatics, and translational R&D. We are also supported by world-class advisors, among them Dr. Alexandra Léry. She's the principal investigator of the TAX2 trial, and she's a leading expert in ovarian cancer at Gustave Roussy Cancer Center, which is the leading cancer center in Europe. Dr. Liliana Schaeffer as well. She's an expert in ECM biology. And our board are seasoned biotech executives and VC representatives. So solid tumors remain really a major unmet medical need, and Apmonia Therapeutics is uniquely positioned to address this with first-in-class ECM targeting peptides. With nearly 17 million euros already secured, we are now inviting private investors to join us in this next phase. So this is your opportunity to support a transformative oncology platform with strong potential for impact and also return on investment. And last, for French residents, also attractive tax incentives. So many thanks for your attention. We welcome your questions in the chat, either in English or in French. So please free to ask.
Thank you very much, Alban. So yes, as Alban just said, you can raise your question in the chat box just below the video normally. Maybe I will jump myself with a first question. In your introduction, you talk about the extracellular matrix, and could you explain us a little more about how it is? So why is this so important regarding to the tumor development?
Oh, maybe I can take this question. Great question, of course. You know, the extracellular matrix has long been regarded as only an inert scaffolding for tissues composed of randomly arranged macromolecules. But now we know that it is absolutely not the case. The extracellular matrix is in fact highly complex, highly dynamic structure network of more than 300 macromolecules surrounding cells and regulating the cell-cell communication in every cellular process within the body. And this extracellular matrix is mainly altered in many ways during the tumor development and the tumor progression at both the primary and the metastatic sites. And we observed numerous, biochemical and also mechanical changes that take place during tumor progression, especially with increased matrix deposition and stiffness and density, thus, creating a physical barrier around the tumor. And this especially shields cancer cells and leads to limit the diffusion and the perfusion of molecules and especially therapeutic drugs. So maybe what can I say to conclude is that targeting the extracellular matrix is probably a key to restoring the immune surveillance and improving also treatment efficacy. And that's exactly what TAX2 does.
Yeah, maybe I can just rephrase in simpler. Most solid tumors have this protective microenvironment, which is made of a dense mass of proteins called the extracellular matrix. And this matrix regulates, for example, the ability of cancer cells to proliferate, their ability to develop resistance to therapies, and also to spread to other tissues to form metastasis. So targeting the ECM is really a promising strategy in oncology, which is still largely unexplored, mostly to technological limitations, in particular in the way to model, to understand the dynamics of these proteins. And this is exactly what we are doing thanks to our discovery engine.
Okay, well, thank you very much. So it raised me another question. So apparently it's a first-in-class approach. So why others haven't think about it before?
Yeah, it's a first in class, as in the sense it is the first to be selective for TSP1 activation of CD47. There are other companies that try in the past to develop CD47 blocking approaches using antibodies, and they generated much excitement in the pharma communities. But these strategies are facing severe side effects that are due to the ubiquitous expression of this receptor, especially on blood cells, red cells, and platelets. So these therapeutic strategies, they promoted severe anemia, thrombocytopenia, and they were put on hold by the regulatory agency, especially the FDA. On the contrary, we are not targeting CD47, but TSP1, which is overexpressed within the tumor stroma. So we got selective tumor addressing of TAX2 and a safer mode of action as well. So in this sense, we are first in class, and we are the first to be selective for TSP1 CD47 axis.
Okay, all right, thank you very much. So then you mentioned you wanted to start with ovarian and pancreatic cancer at first. Why this choice? Well, it starts, I mean, been talking about solid tumor and so on, so it makes some sense, but could you maybe expand a little more about that?
Yeah, we got a strong rationale regarding the role of TSP1 in ovarian cancer. So first, I can say that over 90% of ovarian cancer patients do overexpress TSP1. It's over 80% for pancreatic and also colorectal cancer. And there is a clear rationale for the link between TSP1 expression and prognosis in this indication, and also TSP1 expression and the tumor immune response. And maybe another argument is that there is a rationale in ovarian cancer for developing a companion diagnostic tool based on circulating TSP1, plasma TSP1, in order to be able to stratify the patients that will be eligible for TAX2 therapy.
Okay, yeah, we've got a question going to go beyond the oncology field. Question from Julian, another Julian, where you mentioned that TAX2 could also be used for cardiovascular disease, not just cancer. How advanced are you in your research regarding that topic? And also, if you could mention any other potential peptide that could be good drug candidate.
Yeah, sure. So I can start, and maybe Stéphane, if you want to elaborate after that. So indeed, not only TSP1 is involved in tumor progression, but TSP1 is also released from platelets during the activation step. So we decided to investigate whether TAX2 could be used in other setting, in particular in the field of atherothrombosis and atherosclerosis. And indeed, we got strong animal preclinical data showing that TAX2 is indeed able to delay the thrombosis formation in several animal models, in mice and rats, and also to inhibit platelet aggregation in several species, not only in rodents, but also in dogs. So, and it's also really interesting because not only TAX2 inhibits the thrombus formation, but it does not impact the bleeding, which is the current limitation of existing drugs. And regarding your other compound, indeed, we are also working on other targets from the ECM, I mean other from thrombospondin 1. In particular, we got a product in development which targets cathepsin D. It's an enzyme which is normally only intracellular, but which is released within the extracellular space in some cancer types, in particular in triple negative breast cancer, TNBC. So we are currently doing the preclinical development of this molecule, and we already got some proof of concept in animal models of TNBC.
Stefan, anything to add?
We can't hear you, Stefan.
Yeah, I think you're on mute, Stefan.
But
sorry, nothing to add. The answer was complete.
Okay, well, thank you very much. Maybe a question more related to the business. I think you mentioned that you had already some connection with some big pharma companies. Could you tell us a little more about this, just for us to understand the positioning right now?
Yeah, Abderrahim, you want to start, or
Yes.
you want?
Yes, of course. Can you hear me now?
Yeah.
Yes.
Yes. Okay. Well, we already initiated contact and discussion with several pharmaceutical companies, and they're waiting for the initial data in patients during our clinical trials. We are managing a news flow with all these partners. My colleague Albans mentioned BMS, Merck, Mitsubishi Tanabe, and others. And we used to meet them at least 2 to 3 times each year to share data. And once we have first initial data in phase 1, then we will start the hard discussions in order to close a deal with them once we have the phase 2 clinical trials.
Yeah, we had discussion with them, and basically we clearly know what are their expectations as part of this clinical trial, and we will be careful in documenting the target engagement, the peptide PK in human, of course the safety, and also signs of activity related to the mode of action of TAC. So basically this is what we need to demonstrate before engaging into a licensing deal with these pharma players.
Okay, maybe a question regarding coming back to the science regarding your peptide. Could it be combined with another treatment?
Of course. So the fact that this is totally first in class and that TSP1 CD47 is an axis that is totally different from other immune checkpoint inhibitors, for example, make that TAX2 may easily come on top of other modalities, and in that strong clinical and preclinical data package that support this hypothesis. We demonstrated synergistic effects of TAX2 in combination with anti-PD-1 as an immune checkpoint inhibitor, but also with PARP inhibitors in ovarian cancer model, and also with other targeted therapies such as bevacizumab, and also with chemo. So yeah, we got a wide range of potential combo therapies gathering TAX2 with other modalities.
Okay. And another question about the administration of TAX2. How TAX2 will be administered to patients?
TAX2 will be given IV, weekly IV administration to patient.
Okay, all right. Thank you very much. So maybe a question down to her. I mean, I've got the management team in front of me. So what are the next steps for the next 12 months? Once the deal is closed? Once the round is closed, sorry.
Yeah, so we will file the CTA next month, so the clinical trial application to European regulators in order to be in a position to start clinical trial by later this year and enroll first patient early 2026. So then we will initiate the dose escalation in order to finalize the phase 1 by end of next year and reach the clinical proof of concept by end of 2027.
Okay. Maybe. Thank you. Back to the investment horizon for us as an investor. You mentioned the licensing approach, but what about the pure exit strategy, and how do you envision this?
There are maybe Abderrahim, you want to start, and I can elaborate if you want.
Yes, there are in our shareholder agreement, we have options to allow investors to get what we call preferred
priority dividends.
Yeah. Exactly. One, even with out licensing, you have partial exits of the investors who wants to exercise this, and also usually we are this in the markets. Usually, when you have success, you have two options. After the option after the licensing agreement with pharma companies, go to the markets, to go to public with the IPO, and if we have good windows in the market, and we can expect an exit acquisition of the company by big player, big pharma companies after the proof of concept in patients.
Yeah, one thing which is really important is that people who will invest through capital sale will have access to the exact same shares as the investors within this run, which means that you will have them right, including these priority dividends. So either what will be the partial exit through licensing agreements or an exit through M&A, you will have the same rights as VCs.
Well, good to know, good to hear. Maybe just a quick question regarding all the clinical development that you've planned so far. Right now, focus is mostly Europe, right?
Yeah, indeed. So the first in human, the phase 1/2A trial will be conducted in France and Belgium in the top tier clinical center. So Gustave Roussy in France, Centre Léon Bérard in Lyon in France as well, and Institut Jules Bordet in Brussels, and UZ Ghent in Belgium. This is really excellent centers for early clinical trial in oncology.
Okay, well, I think I don't see any more question. I mean, if you have a burning question, it's now or never. Do not hesitate again to type in the
Just to
question.
add something, if you agree, Julien.
Sure,
with
please.
Alban. Once we have the initial data in patients during the phase 1 trial, we will meet with the FDA in the US to have the pre-IND meeting with them. We will have pre-IND meeting early next year in order to pave the way for further clinical trials in the US. And for this, we will have an office in the US. The company will expand its business activity shortly in short time in Belgium. We will have subsidiary in Belgium, and we will have an office in Boston maybe by the end of 2026. This will allow us to have access to facilities, to incentives in the US for other clinical trials, because we are aware that the major incomes in cancer therapies are in North America.
Well, it's good to conclude this webinar with very interesting and appealing perspectives. So maybe as before giving maybe Alban the last word as a conclusion, I would just remind you that regarding the project, should you be interested by the project, you've got on the top left normally of the screen the QR code that if you scan it, you should land directly on the Capital Cell page. And I've seen that our moderator on the chat has also given the link, so you can click on it. Well, thank you, Benoit. And maybe Alban, because we're arriving to the end of this meeting, and the quick conclusion to summarize the good work, the quality of investment for us, and
Yeah, I can just say one thing. We are an impact company. There are strong unmet needs in this indication, in particular in ovarian cancer. So it's a way to join a financial operation alongside really top-tier VCs. It's a unique opportunity to join and to build a community around this project. So yeah, we are relying on you, and so please feel free to join the run. Thank you.
Well, thank you very much. I think perfect conclusion. Thank you all for having attending this webinar. I hope that you enjoyed it and you learned a lot of things, and you'll be very curious to know more about Apmonia. And looking forward to meet you all soon. Thank you very much. Have a good evening.
Thank you.
Thank you.